Welcome to our video lecture on perception. Our objectives for this lesson, we are going to list different kinds of sensory receptors. We're going to identify some of the structures and functions of the human eye. We're going to talk about, in the human eye, differences between rods and cones. We're going to also talk about the very strange story of the direction of impulses through the nerve cells of the retina. We're going to talk about contralateral processing of the visual stimuli, and then we're going to flip over to ears. We're going to look at structures and functions of the human ear, talk about how the cochlea works, and then we're going to also help ourselves understand how semicircular canals help us maintain our balance. All right, so our essential understanding for today is that living organisms are able to detect changes in the environment. We can detect these changes and then respond to them, and that helps us stay alive. So starting with these different kinds of sensory receptors. So different changes in the environment are going to stimulate the nerves of our sensory receptor organs. There are lots of different kinds of receptors, and so we have mechanoreceptors, which are going to be stimulated by force or movement or sound. So these are literally like things are pushing around. So that mechanical piece of the receptor, so like fingers, skin have lots of mechanoreceptors, and then sound in our ears, of course. Chemoreceptors are going to sense chemical substances. This is super common in our noses and on our tongue. Thermoreceptors sense heat changes, and so again, lots of these on our skin, especially in our hands. And then photoreceptors in our eyes help to notice light. We're going to look just a little bit at the nose and chemoreceptors of the nose. So we have some olfactory, this just means like smell stuff, some olfactory receptor cells in our nose, in the epithelium, or the inside skin part of the nose. These receptor molecules are proteins embedded in the membranes of the cells, and they are coded for by lots of different unique genes. Mice have about 1,000. We don't have quite as many because humans don't smell nearly as well as a lot of other mice. We've not turned on a lot of those genes. And what's interesting is that each cell in the epithelium of the inside of the nose is going to only display one kind of protein receptor. And so each cell kind of notices a different kind of smell and sends that information to the brain. Here, lots of details regarding how these proteins work. These protein receptors are down here, send signals into the olfactory bulb, and then that's going to send signals to the brain. Here are a little bit of a zoomed in picture of those protein receptors that are going to be bound to those molecules that make smells. And what's super cool is that this understanding won the Nobel Prize for Physiology in 2004. So we've really only understood how we smell for a little bit of time, but we do smell with chemicals, chemoreceptors inside our nose that bind to chemical odor molecules, and then that binding of the chemical to the receptor sends a signal to the brain, and this is how we know that we are smelling things. We're going to talk a lot now about the eye. We're going to talk so much about the eye. So this is a diagram of the human eye. Other mammals have very similar eyes. Reptiles and fish have very, very different eyes. But us, we are going to start here. The very outside of the eye is known as the sclera. The sclera is the white part of the eye. In the front, it actually is colorless, and that's the cornea. So the cornea is kind of that colorless bubble over the colored part of your eye, and then the sclera is the white of the rest of our eye. The choroid layer is really just on the inside of our eyeball, and we're going to look at that when we dissect eyes. The fovea is here. This is actually the place where we have the most nerve cells in the whole entire eyeball, and so our best vision actually happens when we focus what we're seeing on the fovea. The blind spot is super close to the fovea, which is kind of weird, but this is where all of the nerves and the vessels of the eyeball kind of merge. And so there aren't any receptors here because instead everything is moving back down the optic nerve, and this will take information to the brain. The retina is the collection of nerve cells along the inside of the eyeball, just inside that choroid layer. Vitreous humor is what fills the whole eyeball. So the eyeball is kind of an empty, I shouldn't say empty, it's a ball, and it's filled with not really tissue, but with this vitreous humor, which is kind of jelly-like. When we dissect our eyes, we'll see that super jelly-like vitreous humor. Conjunctiva, this is just kind of like where the eyeball and the tissue of the face come into contact. Here's that eyelid. Iris is the colored part of our eyes. This is the thing that will either close up a little bit if we have too much sunshine, we don't want too much light getting in, or it might open up more if we need to allow more light to get in. And that light is getting in through the pupil. So the pupil is the hole in the middle of the iris. And what's kind of interesting about that, so we mentioned the cornea here, this colorless part in front of the iris, in front of the lens of the eye. It's also filled with some fluid, but it's a little bit liquidier, not quite as jelly-like as that vitreous humor. And so the aqueous humor is what we're going to call this liquid that's filling up the cornea. Behind the cornea, behind the iris, we do have, I have too many scribbles here, we have the lens. The lens is what focuses light coming in through the eyeball onto the retina. The lens, interestingly, is the only part of the body that doesn't have capillaries. So there are no capillaries feeding the lens. It's not really living tissue. The lens is connected to these couple muscles, the ciliary muscles, and this is what helps to focus when we're looking at things that are close or far. This kind of either stretches or squishes the lens so that we can focus at different distances. So again, we have sclera, choroid, and then retina. In the retina, we have the fovea. This is our best piece of vision where we can see most clearly. This is the blind spot where all of those retina nerve cells and the blood vessels of the eyeball merge and head out to the optic nerve and then to the brain. The eyeball is filled with vitreous humor. The cornea here is filled with aqueous humor. The cornea covers the iris. The iris opens and closes to make the pupil larger or smaller. And then just behind the pupil, we have the lens, which is going to focus all of the light that's passing through the eyeball onto the retina. Pause the video and go ahead and label all the pieces and parts. When you're ready, turn the video back on, and I'm going to label too. All right, hopefully you remembered that this was the sclera, and this is the choroid layer, and then this is the retina, all those nerve cells. We have fovea here. This is our best piece of vision, and then our blind spot is going to be here, and it's because all of the nerve cells of the retina and the blood vessels of the eyeball are going to merge into the optic nerve, which will then send impulses to the brain. The eyeball is filled with vitreous humor. It's a little bit thick. Humor. The cornea is here. This is the cornea. And the cornea is actually filled with aqueous humor. It's not quite as thick as the vitreous humor inside the rest of the eyeball. The colored part of the eye is the iris. It is going to control how large the pupil is. The pupil is the opening. That light will pass through to get into the eyeball. And then here we have the lens. The lens is going to focus this light on the retina so that we can see things. All right, so inside the retina, we're going to focus on the retina now and the photoreceptors that are in the retina. So photo, remember, is all about light. These are cells that receive information about light. So we have two kinds of photoreceptors in our eyeballs. We have rods and cones. Cone, see for color. This is how we're going to remember that cones see color and rods don't. Rods are more about black and white vision. What's cool about rods, though, is that they work really well in dim light. So even just a little bit of light is going to get them to respond. So they're super sensitive to even dim light. They help us see black and white vision, and a lot of nocturnal mammals will have only rods if they're never out in the bright light. they don't have any need to see color. And so a lot of nocturnal animals, mammals, will only have rods, no cones. Cones are super cool. We actually have different kinds of cones. We have cones that see red, we have cones that see blue, and we have cones that see green. And these guys work well only in the bright light. So we really don't see color very well at night because the cones just don't work unless they've got super bright light to be able to turn themselves on and off. And so this electron micrograph picture is showing us rods and cones. The rods are a little bit longer, cones are here, and let's talk about how these guys work. So different wavelengths of light, this is in nanometers of light, are going to stimulate different cones. So we talked about having the blue cone, green cone, red cone. We call them SM and L for some physics reasons, and we can see that the best absorbance of our S cones is in the blue wavelengths of light, and here the green, and here the orangish to reddish. Rods work best kind of in the middle, but they're not stimulated by only green. They can work pretty well across the gamut, but they definitely are still only processing information in black and white. So it's not like they're seeing green. They're not sending information to the brain about green. They're just sending information to the brain about there's light, there are shapes. So color blindness is an absence or a defect of the red or green receptor proteins in those red and green cone cells. What's kind of cool, this gene is on the X chromosome, so this is an X-linked or sex-linked trait. It's a sex-linked recessive trait, and because it is sex-linked and recessive, it's far more common in males than in females. And just a quick little check of your color vision, see if you can see the numbers in these chunks of dots. If you cannot see the 5 or the 8, definitely talk to your doctor about color blindness. All right, so how do these retina cells even work? So we're going to talk a little bit about structure first, and then we'll look at function next. So this is the very back of the eye, so that choroid layer, the very back of the eyeball. And so I know this is not a ball shaped, but here is our iris, here's the pupil, here's the cornea. So light is coming in here through the pupil, through that vitreous humor that fills up the inside of the eyeball, and then it's going to hit the retina. But what's crazy is that the light is going to have to go through the ganglion cells first, and then past some bipolar neurons, and then they're finally going to stimulate cones and rods, and then that information is going to get sent from the rods back to the bipolar neurons, back to the ganglion cells, and then off to the brain. So once again, that light is going to have to come through the eyeball, through all the vitreous humor, through the ganglion cells, past the bipolar cells, until it stimulates either green, red, or blue cones and or the rods. And then these guys are going to send the information back toward the light from the rods and the cones to the bipolar neurons, to the ganglion cells, and then those ganglion cell axons will send the information to the optic nerve and then to the brain. So these eyeball functions are so odd. It is not very efficient. It doesn't make a whole lot of sense. But how these guys work. So our bipolar neurons, these guys in the middle, they are going to send signals from rods and cones to the ganglion cells. But here's where things get crazy sauce. If the rods and cones are not being stimulated by light, for example, if your eyes are closed, what's going to happen is that they are actually going to send impulses. When your eyes are closed and there's no light hitting your rods or cones, these cells are actually going to depolarize and then repolarize, and that's going to release an inhibitory neurotransmitter. So a neurotransmitter is going to be released from rods and cones when there's no light. And this neurotransmitter is actually going to turn off the bipolar neuron cells by making them hyperpolar. So rods and cones will do this, but that's going to send a signal to the bipolar neurons. It's going to be a neurotransmitter that's going to make this part happen. So we don't get this. We only get extra negative. And when these cells are extra negative, they cannot carry out any impulses. So when there's no light, these guys do make neurotransmitters. Those neurotransmitters are going to shut down the bipolar neurons, which means that no signals are sent to the ganglion cells. Weird, right? No light, rods and cones turned on, but turning on the rods and cones turns off the bipolar neurons. So if there is light and these guys are being stimulated, then they actually hyperpolarize. So now the rods and the cones are going to be extra dippy-doo, and they're going to be so negative that they cannot be stimulated even though they're being stimulated by the light. Weird. Anyhow, this is going to turn off the release of those inhibitory neurotransmitters. So we do not have transmitters that are turning off the bipolar neurons, which allows these guys to turn on. So we're going to turn off the rods and cones, and that's going to turn on the bipolar neurons. And then from there, things are normal. The bipolar neurons will release neurotransmitters to the ganglion cells. The ganglion cells will then send signals to the optic nerve and then to the brain, all the way to the back of our brain. Remember, the occipital lobe is in the very back, and that's where we will process all the information that we're seeing in our eyeballs. Weird, but true. Anyhow, so one more little idea about rods and cones. So rods, not super accurate vision, because we'll actually have lots of rods together sending signals to the same bipolar neuron, to the same ganglion cells. We're going to collect a bunch of information and send them to the same cells. So this cell could be turned on, or this cell could be turned on, or rather off, which will turn on this cell, which will turn on this cell. But any one of these three is going to send signals or not send signals to the bipolar neuron that's here. That's different for our cone cells. So each cone cell gets its own bipolar cell, which gets its own ganglion cell. So these guys do not have to share. And so the information that they send is a little bit more specific than the information coming from rods. So these ganglion cells here are going to take all this information. It's going to get sent to the optic nerve, which is then going to send information to the brain. The cell bodies of the ganglion cells are here in the retina. The ganglion cell bodies, the soma of those nerve cells, are here in the retina. And the axons kind of collect into the optic nerve. Crazy, crazy long axons. And again, where all these guys are kind of merging together is a blind spot. So again, capillaries, all those blood vessels, arteries and veins of the eyeball, and then also all of the axons of the nerve cells are going to merge here in that blind spot, which is going to send that information to the optic nerve. So we're actually going to hyperpolarize some of our eyeball cells right now. And so what I want you to do is focus on that dot in the middle and stare at your screen. We're going to stare at the screen for 20 seconds. You can blink, you can definitely blink, but don't look away from the red dot in the middle. So keep looking at the red dot. We're going to look at that red dot for 15 more seconds. You can blink, but don't look away from the red dot. 10 more seconds. Don't look away from the red dot. Blink. Blinking is fine, but don't look away from the red dot. And for 5, don't look away. 4, don't look away. 3, don't look away. 2, oh my gosh, can you see the flag on your screen? Ah, crazy sauce. So we worked so hard to turn off that information about greens and blacks, and we ended up turning on the opposite colors. So crazy neurotransmitter things are going on in your brain. If you're looking, still look at the screen, you should still see red, whites, and blues. Crazy sauce. Super fun trick with your eyeballs. All right, let's talk about how do we even process all this information? How does your brain know what your eyeballs are seeing. So the signals that get sent from the optic nerve to the visual cortex of the brain have some contralateral processing. So the axons of the ganglion cells, we talked about this before, are going to go into the optic nerve and then to the optic chiasma. So this chiasms, anything like that word chiasm, we're talking about things that are crossing. So it's kind of cool. Anything that we see on the right side of either eyeball is going to actually end up on the left side of the brain. Things that we see on, oh, I said right, left, right goes to the left. And then anything that we're seeing on the left side of our visual field from both eyeballs is going to cross in the middle of the brain and end up in the right hemisphere. So things that we see on the left side of our field of vision, left is processed by the right, and then the right is processed by the left. And again, what's kind of crazy, the occipital lobe way in the back of our brain is actually processing this information that's coming from our eyeballs that are in the front part of our faces. And then it crosses, and then it goes all the way back to the back of the brain. Be careful with that word chiasm. Remember that we also have these chiasms here, chiasmata, in crossing over prophase 1 of meiosis 1. Similar word, similar idea. We're talking about things crossing, but we're going to use them in both situations. Good? Good. All right, so now we're going to put the ear, we're going to put the eye away and we'll talk about the ear. So here's a diagram of the human ear, all the pieces and parts that we need to know. The pinna is the outsides of our ears, the parts that we can see. These are pretty good at like catching sound waves and getting those sound waves to flow through the auditory canal, and then those sound waves are going to run into the eardrum, and those sound waves are going to cause the eardrum to vibrate. And so this guy is vibrating because of all the sound waves that are bumping into it. When the eardrum vibrates, it's going to cause these ossicles. Ossicles are little bones. These little bones will vibrate. First, we're going to vibrate this little bone, and then he's going to vibrate, run into this little bone. This guy's going to shake and run into this little bone, and then this little bone is going to shake against what we call the oval window. And the oval window is part of the cochlea, which is in our innermost ear. Also in our inner ear, on the cochlea, we have a round window. We have the auditory nerve, which just like the optic nerve is going to send information to the brain, auditory nerve sending information to the temporal lobes of the brain where our auditory cortex is. And then also part of the inner ear, we have these semicircular canals, and this is what's going to help us with balance. So one more time. What do we have in our ears? Let me start all over with clean slate. So we've got the pinna. The pinna is on the outside of the ear. The pinna is going to lead to the auditory canal. Sound waves travel through the auditory canal and hit the eardrum. The eardrum vibrates, which causes these ossicles to vibrate. We have one, the malleus, the incus, and then the stapes. You don't have to memorize those different names. Just know that there are 3 little bones, 3 ossicles. Ossicles are little bones. That third ossicle, the stapes, is going to vibrate against the oval window, which is going to cause the whole cochlea to do some cool stuff that we're going to talk about in a couple slides. Other parts of the cochlea, we have the round window. We have the auditory nerve, which is going to send information to the brain. And then we also have these semicircular canals, which are all about balance. Good. All right, here's an ear. Go ahead and pause the video, label this ear, and then turn the video back on and check your labels. All right, so hopefully you remembered that this was the pinna, and this is the auditory canal. This is the eardrum. Sometimes we call it the tympanic membrane. Here we have our 3 ossicles, those 3 little bones. This is going to be the oval window. I remember that the ossicles run into the oval window. Those both start with O. The other window is the round one, the round window. This whole purple chunk, this is the cochlea. We have the auditory nerve taking information away from the cochlea to the brain. And then we also have these semicircular canals, and their job is to help make sure that we stay upright. So that balance is coming from semicircular canals. All right, So how does the ear even work? We're going to talk about the middle ear for a little bit first, and then we're going to move into the inner ear. So the middle ear, we've got that eardrum, also sometimes called the tympanic membrane. When sound waves run into it, it's going to shake a little bit. It's going to vibrate. That vibration is going to be transferred to that first ossicle, the malleus, which is going to transfer the vibrations to the second guy, the incus, which is going to transfer the information, that shaking, that vibration, to the third little bone, the stapes. These collectively are called the ossicles, and you just need to know that word ossicle. You don't need to know the three names of the little bones. The ossicles, they're going to vibrate, 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 and then finally they're going to vibrate against the oval window, and that's going to shake some fluid inside the cochlea. We also have this round window, which is super important for like maintaining some balance inside the cochlea. We don't want to shake it so hard that it ruptures. That round window helps to kind of balance things out inside the cochlea. So again, the eardrum, the tympanic membrane is going to shake. It's going to shake against the bones, the ossicles, and then those ossicles are going to transmit those vibrations to the oval window. So I remember O for ossicles, O for oval window. What's kind of cool, this sequence of shaking allows sound to be amplified 20 times. And so even little noises we can hear because the bones can help to amplify that sound. Another super cool thing, though, opposite this idea, if we are near very loud noises, then muscles that are holding, muscles and the ligaments that are holding these bones in places can actually pull the bones away from each other so that they don't vibrate against each other quite so hard and against that oval window quite so hard. So the cochlea, we're going to look at the inner ear now, that cochlea. So we talked about, we've got these, our three little ossicles, that tympanic membrane, the eardrum. Sound waves are going to vibrate, which is going to vibrate this guy, which is going to vibrate this guy, which is going to vibrate this guy, which is going to make that oval window vibrate a little bit. That oval window vibrating is going to shake the fluid that's inside the cochlea here just a little bit. Different wavelengths of sound are going to shake the cochlea at different intensities. What's going to happen is there are these little hairs, these little hair cells inside the cochlea, and when this guy shakes like this, and all that fluid inside the cochlea starts to shake around, these little hairs are going to wave back and forth because of the shaking of the fluid that was caused by the shaking of the oval window, that was caused by the shaking of the ossicles, which was caused by the shaking of that the eardrum. These hairs in the cochlea vibrate, and that's what's going to stimulate nerve impulses that will then send information off to the brain. The hairs in the cochlea actually are wavelength specific. And so the hairs that are here, a little bit closer to the outside of the cochlea, are going to hear like 8,000 hertz. That's big frequency. 4,000 hertz, a little bit closer in, not quite as high a frequency. 2,000 hertz, 1,000 hertz. The closer we get inside, that lower the hertz of the sound, the frequency that we are shaking those nerve cells around. So the hairs will shake at different frequencies of sound, which is going to stimulate different nerve cells to send those sound information pieces to the brain. People who have had hearing loss can actually have cochlear implants, and there will be a microphone put outside their ear, and then a wire is going to be implanted all the way around the cochlea. And when the microphone receives sound waves, it will send signals to different parts of the wire, which will then stimulate different nerve cells in the cochlea, and that will send that same information to the brain that the hair cells were sending. amazing. So these nerve impulses from the cochlea are going to be transmitted to the auditory nerve. Auditory nerve here is going to send information to the temporal lobe where the auditory cortex is. So those cochlea hair cells will depolarize. They will send some neurotransmitters to the sensory neurons in the auditory nerve, which will again send that impulse to the brain, to the auditory cortex, which is interestingly super close to our ears, as opposed to when we're doing eyeball things, eyeballs here processed by occipital lobe way back here. And last but not least, we're going to talk about these semicircular canals in the inner ear. So cochlea that does the hearing part was here. This chunk is going to be all about balance. Sometimes we call it proprioception, or the vestibular system, and it's all about just keeping upright. So there are 3 different semicircular canals, and what's kind of cool about these circular canals, they are oriented in X, Y, and Z 3-dimensional arrangements. So we have some that go up and down, some that go sideways, and then some that go like in and out of your screen. And these guys are also filled with some little hair cells and lots of fluid. And when we tilt our head, fluid is going to move through and past these hair cells, which is going to cause those hair cells to wave over, which is going to send signals to the brain that says, hey, you're sideways. And then if we pick our heads back up, that fluid will kind of go back and the hair cells will go back this way, and then the brain will be like, we're oriented to the right direction again. So we have these, again, in all 3 of those orientations, our X, our Y, and our Z. And this is just what helps us with keeping ourselves upright. Super cool. All right, and hopefully we achieved all of these objectives. We talked about those different kinds of sensory receptors: mechanoreceptors, chemoreceptors, photoreceptors, thermoreceptors. We talked about all those different cool parts of the human eye. We talked about how rods and cones work similarly and differently. Cones for color, and then rods are not color, but they help us see better in the dark. We talked about the direction of impulses through the nerve cells of the retina. Remember that we've got those rods and cones in the very back, and they're going to, when there's no light, send a signal to the bipolar neurons, which is going to turn them off. But if there is light, then these guys get turned off, which turns these guys on, and then they'll send a signal to those ganglion cells, which will then send signals to the optic nerve, which will send signals to the brain. We talked about contralateral processing of visual stimuli, things that we see on the left side. This eyeball right here that's on the left side of your screen is actually being processed by the right side of your occipital lobe right now. We talked about different chunks of the ear and how the cochlea works, that there are little hair cells that are stimulated by specific wavelengths of sound waves. We talked about the semicircular canals that also have some hair cells that are going to help us keep ourselves upright. And that, my friends, is the end of our lecture. Good job.